Today we're going to explore a technique called electroporation, which plays a key role in the delivery of gene therapy. Gene therapy overview. Gene therapy aims to treat or prevent disease by modifying or manipulating genes. This can involve replacing, inactivating, or introducing new genes to avoid genetic disorders or diseases. One of the promising methods being studied for delivering gene therapy is electroporation. What is electroporation? Electroporation involves by applying an electrical field to cells. We crack temporal pores in the cell membrane. The process starts with the application for our short electrical pulse to the cells. This route to the cell lipid bilayer. As a result, pores from in the cell membrane allowing genetic material such as DNA or RNA to enter the cells. Once the electric pulse is removed, the pore reseal and the seal retains the introducing genetic material. The genetic material can be integrated into the cell's internal machines, enabling it to carry out its therapeutic functions.
Now let's discuss the main characters of electroporation using non-viral vectors. This technique uses plasmid or RNA instead of viral vectors, avoiding the risks associated with viruses, such as an immune response and intersectional mutagenesis. In terms of efficiency, it allows for the delivery of large DNA fragments compared to viral vectors, which is particularly useful in studies requiring the insertion of large genes. It is also highly versatile as it can be applied to a wide range of cell types, including primary cells, stem cells, and immune cells that are usually difficult to transfect. Another key feature is the precise control over the amount of genetic material introduced, which allows for adjusting expression levels according to the study needs. Moreover, extrapolation offers the possibility of transient transfection, temporary gene expression, as stable transfection, gene integration into the genome, making adaptable to different experience goals. Let's move on the advantage of this technique. Regarding safety, since it doesn't involve viruses, the risk of intentional mutagenesis is significantly reduced, which is crucial for clinical application. It also very flexible, easily adaptable to different cell types and tissues, making it useful for both research and therapeutic application, such as genetic modification of immune cells for advanced therapies. Additional, since it doesn't rely on viral proteins, the technique poses a lower risk of immune response, making it safer for use in humans. Electroporation is also cost effective as producing non-viral vectors and using electroporation. Equipment are generally less expensive and the equipment is reusable, which reduces long-term operation costs. In terms of scalability, the technique is suitable for both laboratory experiment and clinical application, making it useful for different contexts from basic research to the production of gene therapies. Finally, the precise control over the transfection process allows for adjusting the electrical pulse parameter, thereby optimizing efficiency while minimizing cell damage.
Now, continuing talking about the disadvantages of electroporation. While electroporation can introduce genes into the cell, its efficiency is often lower compared to viral vectors, particularly in hard to transfer cells like primary neurons or stem cells. For example, viral vectors often integrate more efficiently into the genome, whereas electroporation often requires optimization to achieve comparable results. When we talk about optimization, we are talking about the cell viability concerns. In this therapy, we have to use electric pulses that can cause stress or even cell death if the parameters like voltage or the duration of the therapy aren't carefully controlled. And lastly, transient expression means that for chronic conditions, electroporation sometimes doesn't work because the effects may wear off. So it's a challenge for chronic conditions where long-term gene expression is needed. To wrap up everything that was said today in the video, we know that electroporation is a very important tool in gene therapy with specific advantages like non-viral delivery. However, its limitations, such as efficiency and transient gene, mean it is often used in combination with other techniques. We know that future improvements will optimize its parameters and could further enhance its viability in clinical settings. Thank you.